An engineered nanomaterial, or ENM, is any man-made particle with one dimension less than 100 nanometers. There is great interest in the many potential benefits of ENM in our lives, but there is concern that some ENM may cause adverse human health effects. Research is being conducted to determine what properties of ENM may cause adverse human health effects. In order to study ENM health effects, scientists either instill a specially prepared ENM suspension or expose rodents to ENM in air. This animation presents a proposed model of events to illustrate our current understanding of how exposure to certain ENM may progress to inflammation and lung disease. Creating ENM suspensions. ENM are added to a dispersion medium containing lipids and proteins that are similar to what is found in the lung airspace. Because ENM are so small, strong forces hold them together to form large agglomerates. Lipids and proteins adhere to available ENM surfaces, forming a corona, and are necessary to improve dispersion. Acoustic energy helps to form smaller agglomerates. Lipids and proteins adhere to newly exposed ENM surfaces, forming a corona. Inhalation exposure of ENM is an area of concern. Because of their small size, ENM, either instilled or inhaled, travel through the bronchioles and reach the alveoli, where gas exchange takes place. The alveoli are covered by a thin lipid and protein layer called surfactant. Under the surfactant layer is a thin aqueous layer containing more proteins and cells. ENM penetrates the surfactant layer, reaching the aqueous layer. The final ENM corona, regardless of the nature of exposure, should be similar and reflect the makeup of the lung environment. Macrophages in the aqueous layer are responsible for removing inhaled materials. Macrophages recognize the ENM as foreign material to be removed through phagocytosis. The ingested ENM are engulfed in phagosomes. The phagosome is now in the cytoplasm of the macrophage. In the cytoplasm, the phagosome containing the ENM comes into contact with a lysosome containing hydrolytic enzymes. The phagosome and lysosome merge to form a phagolysosome. The hydrolytic enzymes in the phagolysosome break down the ENM corona, but do not degrade the ENM. The exposed surface of the ENM is now free to bind to the inner membrane of the phagolysosome, disrupting the normal membrane organization. The disrupted membrane organization causes pores to form in the phagolysosomal membrane, allowing degradative enzymes to escape into the cytoplasm. One of the enzymes, cathepsin B, initiates the assembly of the NLRP3 inflammasome. The assembled NLRP3 inflammasome activates cleavage of inactive procaspase 1 to active caspase 1. The active caspase 1 cleaves pro-IL-1 beta into IL-1 beta. IL-1 beta is released from the macrophage, initiating important first steps in lung inflammation. Additional lysosomal enzymes are released into the macrophage's cytoplasm. These lysosomal enzymes initiate many other damaging effects, an example of which is cleaving BID into TBID. TBID binds to the surface of mitochondria. This process causes pores to develop in the mitochondrial membrane, generating the production and release of ROS into the cytoplasm, causing damage to the cell. Taken together, ROS, lysosomal enzymes, and active caspases in the cytoplasm cause irreversible damage, leading to macrophage death. Most of the ENM are not cleared efficiently because of macrophage injury. Consequently, the entire process continues to cycle, with the death of macrophages and the release of IL-1 beta contributing to chronic inflammation. The cycle is similar to what occurs during exposure to other known inflammatory particles and fibers, such as crystalline silica, silicosis, asbestos fiber, asbestosis, and even particles that are endogenously produced, uric acid, cholesterol, and amyloid plaques, leading to a number of human diseases. Improved understanding of this pathway will lead to selection of safer E&M and to new therapies to treat human diseases caused by pore formation in lysosomes.